Hello students, let's discuss now of cell membrane. Cell membrane is also known as biomembrane. They are also known as the plasma lemma, plasma membrane. This is because different scientists have given different terms. The plasma membrane term was given by Pfeiffer. Likewise, plasma lemma was given by Flower. And the biomembrane was the term which was given by the Singer and Nicolson, who were those proposed fluid mosaic model as well. So biomembrane is given by the Singer and Nicolson, and likewise unit membrane was the term given by the Robertson. Robertson was a scientist who gave it the term unit membrane. Cell wall is a thin, elastic, living, selectively or differentially permeable membrane. Cell membrane is made up of phospholipid and protein. Phospholipid it consists of head and tail. The head here is polar. Head is polar, that is, it is hydrophilic, that is, it is water loving, and hydrophobic, that is water hating, and the tail is non-polar. As phospholipid has both property of hydrophilic as well as hydrophobic in one, they are known as the amphipathic molecule. Phospholipid is the amphipathic molecule. Selectively permeable, semi-permeable, and in general, plasma membrane with the organelle membrane is known as the biological membrane. Phospholipid in membrane is always bilayered. In case of membrane, the phospholipid is always bilayered. And there are various models to explain the plasma membrane. There are one, that is the lipid membrane model, also known as the lipid bilayer model, which is the model given by the Gotter and Grandson in 1925 this model was given by them. Before it, in 1895, Robertson found that fat-soluble substances passes easily through the cell membrane. He therefore concluded that the cell membrane contained lipid. As the fat-soluble substances were passing, and the existence of the lipid layer was confirmed on the basis of experiment on wetting the cell membrane with oil. A drop of oil was placed in contact with the cell, spread over and adhered to the cell surface. In 1895, Robertson found this. And later the Gotter and Grendel in 1925 were the first to suggest the possible structure of cell membrane. They were the first to suggest possible structure of the cell membrane, and they studied RBC and found that the lipid extracted from the RBC's membrane were suspended in water, and they found that the lipid extracted from the RBC's membrane after suspending in water is spread as a monolayer at the air water, and the air occupied by this monolayer was nearly twice the total surface of the intake RBC. And hence they concluded that the plasma membrane consists bilayer, and the polar group of these molecules were situated outside of the layer. And this is the lipid bilayer model was given by Gotter and Grendel. They studied the RBC. And next model is the model given by Daniel and Davson, which is also known as the sandwich model, that is also known as the protein lipid protein hypothesis. Sandwich model as the lipid, it is the, let me discuss with the detail, that look at the Davil and Davison model. At the upper point there was protein, that is the globular protein and the absorbed protein. They attempted first to describe the membrane structure in terms of molecules. They were the first to suggest, and they were the first to describe the membrane structure in terms of molecules and to relate the structure to the biological and chemical properties. According to biomolecular model, plasma membrane consists of the two layers of phospholipid molecules, where the molecules of the phospholipid are arranged in such a way that hydrophilic heads, that is the polar part, is facing outside, and the hydrophobic, that is the non-polar part, is facing, are associated in the regions of the leaflet. It is in the middle, as is the inner region of the leaflet like model, and this model postulates the presence of both lipid and protein in the membrane. The hypothesis also suggests that the polar ends of the lipid molecules are associated with the monomolecular layer of globular proteins. The main drawback is that it does not explain the function and the phenomenon of active transport movement. So this model was not accepted. This model does not explain the phenomenon and the function of active transport movement. So this model was not accepted. And the next model is the unit membrane model. This was given by the Robertson, J. David Robertson, on 1953, and it was the protein lipid bilayer protein structure. He forwarded his unit membrane hypothesis, in which he stated that all cellular membrane has a similar trilamellar structure that consists of lipid bilayer, this is the lipid bilayer. It consists of the lipid bilayer between the two layers of protein. He modified the sandwich model given by Daniel and Davson, that there is the difference in the protein that is present on the outer and the inner layers, that is, on the outer protein part it is made up of mucoprotein, and in the inner part it is of non-mucoid protein. So this was only the difference between the Daniel and Davson model and unit membrane model. That is, he was the one to extend about the difference in the protein of outer and inner layer. And the last model is the fluid mosaic model, which was proposed by the Singer and Nicolson, who gave the term biomembrane also. And in 1972 this was found as the most recent convincing model. And according to this model, plasma membrane is a tri-layered structure that composed of lipid, protein and some amount of carbohydrate. The membrane does not have uniform